Hello everyone, my name is Ashfaq Ahmed and I work in the field of bioinformatics and structural biology. Today we will learn how to calculate genetic diversity, segregation, segregating sites, haplotypes, haplotypes frequency, and finally we will learn how to construct a haplotype networks. During this session, I will also provide feedback on basic concepts from evolutionary biology and biology. This session is very fruitful for those people who are working in the field of DNA barcoding. And in my experience, most of these guys are from plant sciences. So this video is best. This video will not only teach you how to do these things, how to do these analysis in just a kind of 30 minutes. Also, it a publication quality images for you. So let's start the video. Okay, first of all, you have to open Google Chrome. I'm using Google Chrome. Whatever you are using, that's up to you. And type collab.research.google.com/github github/h100. This is my repository if you are new to this channel, so it will be your first time to experience working on the Colab or in my repository. So welcome again. And if you are unable to spot this link, so please look to the description section of this video. This link will be available there. Okay, after typing this link, just click enter. You will see this kind of dashboard or window, whatever you say. Click here and come select plant sciences. Now you see genetic diversity, segregating sites, haplotypes, and networking. Click this option. Okay. Now click this option, connect. Wait for some time, possibly 30 seconds or less. Okay. This is a general introduction of the notebook. Like I write this notebook, this is my introduction, and this notebook can be used for research and teaching purposes. Okay. If you want to know my channel or the video link of this notebook, so you have to click here. Okay, this is the content. Like we will learn how to calculate nucleotide diversity, segregating sites or gene segregation, haplotypes or haplotypes networking. But to dig down, we have to understand briefly these terminologies. What are nucleotide diversity? Nucleotide diversity is actually a molecular genetics concept which actually measures or which actually calculates the amount of genetic variations or the amount of base pair differences in a population. From the population, I mean the dataset of the sequences you exported to this notebook. For example, you have a data set of alignment sequences, whatever you got it from NCBI, BLAST, that's up to you. But one thing, it should be in aligned form. I mean, it should be aligned with Cluster W or any other software of your choice. Okay, then we will calculate the nucleotide diversity first. And I told you the nucleotide diversity is not just the measure of the amount of variations or dissimilarity or the diversity between that set of population or between that set of sequences. Okay, the next thing is segregation. The process by which individual genes are segregated and passed down from parents to offspring. So now you have this data set of from plants, from animals. We will not discuss that. But on the basic definition, if two sequences they're sharing the same, how to say, evolutionary ancestor, but they are different currently. So possibly or most probably they are segregating from the same ancestor. It means the simplest way I could explain was like that. Okay. Then what are the segregating sites? So if you understand the segregation, then you will understand the segregating sites. If two sequences they are separating and they are emerging from a same ancestor, but they have some differences. So the differences will be present at the sequence level. So the sequence level or the sequence differences, those base pairs, which segregates these two, which differentiate these two sequences in a phylogenetic tree or anything else, we call it getting sites. And then the next thing is haplotypes or haplotypes frequency, or you can also say haplotypes distribution. So what haplotype? Haplotype indicates a group of genes, a group of genes which you have in your data set or a DNA variants. Once again, you have all these things in your data sets. What is your data set? A sequenced alignment which you got it from somewhere. So a group of sequences are a group of genes that are inherited together from a single parent. So we will call it haplotype. For example, in the short simplest form, for example, you have 10 sequences, and when you cluster them based on the nucleotide diversity and segregation, so you may receive three clusters. In one cluster or in one group, you will have three sequences most similar to each other. In another group, you will have three more similar to each other. And in the third group, you will have four more sequences similar to each other. So basically, these three groups can be called haplotypes. And what are haplotypes network? So at times we network them to see the association of this group to another group and to decide which group is evolutionary near to another group. So these are all those things. So now we are going to start the real business. We will use BioPython and we will need FASTA sequence files or dataset which contains FASTA sequences. So first of all, you can see your notebook is connected. Now just click here. It will, and you will see this kind of thing. So just run away anyway, and it will take almost 10 to 15 seconds. With me, it took 5 seconds. Okay. What it did, install the BioPython to this notebook. So once you see this kind of thing, this tick mark, that means the installation of BioPython is complete. Now we will need, or this notebook will need, your FASTA sequence. So your FASTA sequence should be in the FASTA format. If you don't know how to do it, it's very easy. If you have any program, let's say I'm opening this BioEdit. This one, open, if you have sequences, just open those sequences. Let's say this is align sequences. Click here, accessory applications, and then wait. Now you can see here accessory applications. Then use cluster multiple W alignment and then run this cluster W. It will generate the alignment, although it's already aligned, and then click here file and click here save as. It will save the file by any name you want and anywhere you want. Just select the FASTA save as type here. So it will select your sequence dataset in a FASTA. Uh, Now, to do that, what you need, just click here. It will run this cell and it will ask you to provide your FASTA file or provide your dataset. It's okay. So I think the my internet has some issue, so it's it's not initiating. So just wait. Okay, I clicked it again. Once you click it, you will see this kind of thing: choose files or cancel upload. So you have to click the choose files, and from the choose files, I will just go to some. I will just upload some random data here. So let me think. Okay, let me upload some render data from here. Okay, so can you see one tRNA sequence is that fast? So I'm uploading this dataset. So you will upload your own dataset here because I'm not providing any dataset with this tutorial. Okay, now after clicking this, it will import some necessary libraries to the space. Okay, now we are going to calculate the nucleotide diversity or PI. Just click here. It is calculated. Sorry, here we just set the functions. Now here the functions are ready. We are going to calculate the nucleotide diversity. Just click here. Okay. See the answer. The nucleotide diversity is 0.011 whatever in your case. So this is your nucleotide diversity. Now, successfully we have calculated the nucleotide diversity for our dataset. Now we are going to calculate the segregation and then the segregating sites. We will also visualize the segregating site. So just click here, it will calculate the segregating sites. So in this data, it is saying that 21 segregating sites are present. Okay. Now I want to visualize them. What are those 21 sites? So click here. It will take a while and then it will generate a very beautiful heat map. And this heat map, these are the segregating sites and these are the positions in alignment. So remember, the position in alignment are a little different from the real sequences because in alignment you are also seeing the gaps. So that's why I dropped it position in alignment. So if you want to mention the real alignment or the real position of the sequences, then you have to match it with the alignment position of that nucleotide to the database, and from there you have to write or to uh, edit this picture. Now if you don't know, this picture is an and this is the legend, kind of legend that tells you that if it is yellow, so it means 100% confident, with confidence, that this is a segregating site. Now if you want to use this picture in your manuscript or thesis, how you will get it? So simple. I have this script has already downloaded. You can see segregating site. Now click here and then download. After some time, it will download this picture. Now let's see, is it downloaded or not? Double click. Now you can see, and it's in the 600 dpi, extremely high resolution. Even if we are going to uh, zoom it, you can see still the point 4 is clear. So this is your first picture, and you have calculated the nucleotide diversity. Then next you calculated the segregation, and then you plot the segregation sites in your data. Now the next thing is we are going to calculate the haplotype diversity, or you can also say the haplotype frequency. So the haplotype diversity or the haplotype frequency is 0.836. Now, but this is a statistical or mathematical value. I know as a researcher you are interested to see those haplotypes or to plot those haplotypes. So for that you have to run this cell. Can you see? Okay. Okay, it will generate the same thing again, but the mapping will be created here. Just run here. Now, you will see all the haplotypes of your datasets. How many? 1, 2, 3, 4, 5, 6, 7, 8 haplotypes. You can count them here. Now, haplotype 7 is the most with 28.6% distribution or 28.6% frequency. So it's up to you what to call it. Now, another important thing I have added in the code, if you want to know what members or what sequences are maintained by haplotype 7. So just scroll down, you can see these are the two members of the haplotype 1, these are the one member in the haplotypes 2, 3, and the haplotype 7 contains 4 members. Okay, so now you can copy this information with yourself in any text files. It will be uh, during writing of your thesis or manuscript research papers, you will need these information. But if you are interested in this figure, so don't worry, once again, it is generated here, haplotypes frequency. Click here, download. Can you see? Now, whatever you want, where you want to use it, just you can use it. Okay. Now, that's done. Until now, we have successfully calculated the genetic diversity, segregation sites, we have successfully calculated the haplotypes and their frequencies. Now we are proceeding to the network of those haplotypes and elucidate some meaningful association. We will use network X, it's a program, and matplot for plotting. To further clarify like how this step will take place, so actually we are calculating Hamming distance. If you don't know Hamming distance, or let me tell you, in bioinformatics we have two types of distances: basic distances, Levenshtein distance, and Hamming distance. We calculate based on the genetic diversity or based on the genetic sequences variations or differences. We calculate the distance between two sequences present in the dataset. From the two sequences, I mean between the every two sequences present in the datasets. And then based on those distances, we create a haplotypes. So sequences containing or sequences mostly closely related to each other will be grouped into one haplotype. Sequences far away from other sequences will be grouped into another haplotypes, which we have done already. So now we will network these 8 haplotypes to see their association. How we can do it? Just click here. It has calculated some networking thing. Now we will run this and it will generate kind of network for you. You can see these are your 8 haplotypes, haplotype 6, 3, 8, 5, 2, etc. etc. These are the legends of the haplotypes. And what are content? The haplotype contains 4 members. I already mentioned to you above. So you know now, like, what are those 4 members? Now, the final thing I want to teach you that you are seeing some crosses or some edges. We call it edges, and these are called nodes in the language of bioinformatics. So, if you want to see haplotype 6 and haplotype 1, they are connecting each other with a value of 16. But if you see haplotype 6 with haplotype 7, it is connecting with the value of 2 here. That means the 2 value means that the distance between haplotype 6 and 7 is smaller compared to the distance between haplotype 6 and 1, which is 16. So now it's a common sense that the sequences present in the haplotype 1 is far away present or is far away in the evolution from the sequences present in haplotype 6. But the sequences present in the haplotype 6 are near to the sequences present in the haplotype 7 because the genetic distance is 2. The higher the number, it means they are far away from each other. And the smaller the number, it means they are near to each other. I hope I have explained each and everything for you in just 23 minutes. So, and I tried to speak faster to make this video shorter. So, once again, congratulations to all of you. You have learned something new in the most easiest way possible. If you want to keep learning with me, please support my work by subscribing and sharing this work or this channel. Okay, thank you so much. If you have any question, you can drop your question in the comment section. Bye bye for now. Thank you.